Thank you, Neha, and thank you, Professor Kozlowski, for this wonderful lecture. Moving on to the Q&A session. Uh, the first question is it possible to use AI or ML models, design such catalysts, and predict the reactions, yields, or products?
Yes, it is. That's one of the areas that we're actively exploring. So in the first topic that I showed you, where we're generating these large data sets, we're now trying to use them to predict outcomes. And we have some promising results, but we still need to collect more data to test the model and whether it works or not.
Sure. Thank you for your response. Moving on to the next question. What are your views on carrying out the phenolic coupling electrochemically?
Electrochemical phenol coupling has been well explored by Siegfried Waldvogel in Germany. So it's not something that we've looked at ourselves. Early on, before he had published much of his work, we had thought about doing it with a copper catalyst in electrochemistry, but we had difficulties in making that work. Using metal catalysts in combination with electrochemistry does take a bit of expertise that we did not have, but it can be effective.
Thank you. Moving to the next question. In what scales were the reactions carried out, and does the results with high throughput experiments correlate with the experimental yields when the reactions are scaled up, for example, 0.3 or 0.6 millimole?
Yeah, so the high throughput experiments are run on very, very small scales, usually 2 to 5 micromoles. That's nice because you don't need a lot of substrate to be able to do a screen, and you can get a lot of data fast. But you do bring up a valid point, or this person has brought up a valid point, is do the reactions still work when you scale them up? Because there are many artifacts that can be introduced in high throughput experimentation. So the first thing we do when we get a set of results from high throughput experimentation is we validate on bench scale. So we take 5 or 6 of the best results and we rerun them on a larger scale reaction to see if it still works. And I'd say roughly 80% of the time it does. But every once in a while, we're unable to reproduce the result from high throughput experimentation easily. And so then we have to go back and we have to scientifically analyze what's different between the small scale and the large scale process. One example is in high throughput scale, we layer everything down with solutions. So we'll put first the catalyst in a solvent, and then we'll evaporate that solvent. And then we'll introduce our additives and we'll evaporate that solvent. And then we'll introduce our substrates and then we'll add the reaction solvent. Some of those trace solvents left behind can actually play a role in the reaction. And so that's something that can change the outcomes. And if we reproduce that on a large scale, we can get the same results. But there are a host of other reasons why a high throughput reaction might not necessarily scale well. Another interesting one we found is that we use internal standards in our high throughput reactions. And in some cases, when we remove those internal standards on scale, the reactions don't work. And the reason for that is that these were photocatalytic processes. The internal standards were large aromatics that prevented back electron transfer. So validation from high throughput is extremely important in moving a project forward.
Thank you for the detailed explanation. The next question. Have you carried out any kind of DFT calculation for those catalytic reactions?
Yes, so for most of the mechanisms that I showed in this talk, we have done DFT calculations to assist us in understanding the mechanism. We also had done experiments to establish the molecularity, the sequence of steps, the rate limiting steps, and then we used calculations to help with the steps that we cannot probe directly by experiment. That's when it's the most powerful. And the cross coupling that I showed, it was the step after the rate limiting step is the carbon-carbon bond forming event, and it's very difficult to probe that experimentally. And so we used computation to understand what was going on there.
Thank you. The next question, Does the oxidative coupling reaction work with electron withdrawing substituents on the benzene ring?
Yes, so that is one of the limitations of what I've shown today, at least, whether the reaction will proceed with electron withdrawing substituents on the aromatic ring or not. Typically for the processes that I've shown today, it does not. However, I'm pleased to say that we've developed a new process where we can tolerate electron withdrawing substituents, including esters, nitriles, and so far one nitro substituent, which is going to be a real big advance. That's not quite ready for me to share with you yet, but that should be coming out by the end of the summer, I believe.
So you're looking forward to it. So next question. Why was the second CA's amination low yielding?
Yeah, so this is in the photocatalytic coupling where we did first an SNAR, which we found is typically a much higher yielding process. The second oxidative amination, we're not entirely sure why that doesn't work as well. One interesting thing is that we're able to use heterocycles that are actually quite oxidizable. So that's interesting that we can get that selectivity because those other heterocycles do not form the donor acceptor complexes very readily. The donor acceptor complexes form between the pyridinium and the phenol. We believe that's driven by hydrogen bonding. And that the efficiency, once we get that radical cation, is just not as high with those nucleophilic heterocycles in that oxidative process. And that perhaps there's some reversibility there. So you make the nitrogen carbon bond, but then you can break it. You make it, you break it, and every once in a while it oxidizes a little bit further, and that's a product.
Thank you. The next question. Does the coupling reaction work with quinoline and its derivatives?
Quinoline.
Yes.
Okay, so all the couplings I showed between heterocycles and phenols, they're with 5-membered heterocycles. And the reason for that is 5-membered heterocycles are nucleophilic. 6-membered heterocycles typically are not nucleophilic, they're electrophilic. And so they would not be an effective reaction partner for a phenol, at least in the way that I've shown here. But that is something that we are very interested in looking at. We would have to do an umpolung, though. We would have to use the phenol as a nucleophile and the quinoline as an electrophile. And that's another set of projects that we're exploring, and we have mixed results. In some cases, we can see good reactivity, but we haven't been able to find something that's broadly applicable to a range of 6-membered nitrogen-containing heterocycles.
Thank you for your response. Moving to the next question. Could you please clarify why manganese is more favorable for the ortho-para coupling?
Okay, so chromium is really good at the ortho coupling too. Or sorry, why does the ortho-para coupling?
Yes.
Yeah, I have to say that we haven't studied every single catalyst we looked at to figure out the mechanisms. We've studied the chromium in depth, and we've studied the vanadium and copper in depth. Manganese is one that I would like to go back to and understand a bit more as to why we see this difference. So I don't have a full understanding of that yet. I would have thought that manganese would react very similarly to chromium. So there is something very subtle going on here that's not easy for me to explain or understand at this time. That's a good point.
Thank you. The next question is more related to organic letters. What sort of heterogeneous catalysis falls within the scope of organic letters?
Yeah, so the question is, what sort of heterogeneous catalysis falls within the scope of organic letters?
Yes.
So you need to keep in mind that organic letters audience is organic chemists, right? And they are less interested in the development of heterogeneous catalysts. So, for example, making different kinds of zeolites or different sorts of polyaromatic materials, unless you can show that they give new reactivity within the context of an organic transformation. If that's not the case, then that work is probably better suited to a catalysis journal or a solid state catalyst journal of some type, of which there are some in the ACS portfolio. And our goal as publishers is always to get your science in front of the people that care about it the most. And sometimes, I've had my work rejected from organic letters, just like many people have. Sometimes that happens not because the work isn't good enough, but because it's not the right audience, right? That I haven't picked the right journal for my work. And that's a really important component when you're thinking about publishing, is where to publish. And for organic letters, we're interested in novelty on the organic chemistry portion, and particularly for heterogeneous catalysts that are doing transformations that can be undertaken successfully with many standard reagents that we can purchase commercially. That really isn't a good fit for organic letters.
Thank you. Moving to the final question for the day. Can the protocol be applied to aniline couplings?
Yes, we've published on that. We've done cross couplings of anilines with phenols, and we published on that in our organic letters. That was done with Tom Paniak, another graduate student in the group.
Thank you. That brings us to the end of the Q&A session. Over to you, Neha. Thank you, Professor Kozlowski, for the responses.
Thank you so
Thanks for
much.
moderating.
Yeah.
Okay.
Thank you so much, Professor Kozlowski. It was a wonderful question and answer session, and thank you, Manas. So I would just like to request you for some concluding remarks for our audience.
So first of all, once again, it's a pleasure to see so many people from so many parts of the world engaging in science and joining this session. It was a real pleasure for me to share that with you. And then the final remark I have is I hope that you're all productive in your science, and I hope that you think of publishing in organic letters if you have work that is a good match to our scope. And I would look forward to receiving your submissions. And other than that, I wish you all the best success in your research.
Thank you so much, Professor Kozlowski. So this brings us to the end of today's session. We hope that you enjoyed this broadcast, and we invite you to view the editorial recordings for the past events in the ACS Science Talks library. Professor Kozlowski, it would be great if you can please stop sharing your slides. Thank you so much. Okay, so for the attendees, you will receive your certificate of participation along with a survey form on your experience about the ACS Science Talks. And on behalf of all of us at ACS, thank you for joining us today. We hope to see you again soon for more ACS talks and events. Until then, stay safe and healthy. And again, Professor Kozlowski, thank you so much for joining in. I understand it's quite early for you today to present, but thank you so much.
Thanks.
Thank you so much. Have a great day.